Good afternoon, everybody. It's a pleasure to be here with Professor Alina Popp and Professor Valentina Discepolo. The webinar of this afternoon will be on drug therapy for celiac disease. There will be two talks. The first one from Alina Popp from the University of Bucharest, Romania. She is a very important expert in the field of celiac disease, and particularly in the analysis of histology and new technique to evaluate histology in celiac disease. And after Valentina Discepolo, that she is from our university in Naples, she's associated professor in Naples, and she will talk about new therapy and new trials that are going on in the field of celiac disease. So please, Alina, if you want to share your screen.
Thank you, Renata, and good afternoon to all. It's already dark over here, so it might be better to say good evening to everybody. I'm really very happy. I'm happy I was invited to give this talk, and I will soon start to share with you what was, let's see, the emerging main problem, or let's say problems and issue during the work of a task force. This was just a moment. Do you see my screen now? Because I started, yes?
Yes.
Now it's moving.
Yes.
So the issue was, in a way, we thought it would be good to have a joint venture between ESPGHAN with NASPGHAN together in order to produce a position paper regarding novel therapies in children for celiac disease. And this was from the ESPGHAN part, this was led by Riccardo Troncone from Napoli group. And as we now speak, let's see, this has difficulties to move on. Yes. But we should really start by saying that the gluten-free diet is the only available treatment in children as well as adults. And this is a very clear fact, even if there are a lot of heavy, strong research in the drug development. So when thinking of the recent concepts and recent very new knowledge on drug therapies, it is most probably coming, maybe soon to come, drug trials or opportunity for drug trials in children. So this is why we felt the need of looking a little bit more carefully into it. And let's go to the whys. I mean, why novel therapies would be needed? And of course, first of all, is these difficulties in really avoiding the gluten, really avoiding the inadvertent gluten ingestion. Many children of certain age, especially, they have difficulties to really keep the gluten-free diet, and this is coming with a lot of long-term negative health effects as a clear and direct consequence, is especially on, let's say, long-term inadvertent gluten ingestion. And we all have our own patient, and we know in clinical practice that not all children really heal. Some of them, they have persistence of symptoms, and these symptoms are sometimes underlined with the persistence of mucosal injury. So what to do with these ones? And when it comes to novel therapies and drug trials, of course, we have to keep in mind the safety and the ethical consideration, and which one would be the best target group or groups of children, and how to design the study and how to choose the best outcomes for the study. Well, first, when thinking about safety, we really know that we cannot simply just extrapolate the data from adults drug trials in many of chronic diseases, not only in gastroenterology, because it might be so that the dose response would be different, or the drug metabolism could be different. So it is really so that at least not all the phases could just simply be extrapolated. So we really need drug trials especially designed for pediatric population. And in celiac disease, this is when looking into the studies of adults, we really know that this would mean, at least for certain type of proposed therapies, this would really mean the gluten challenge. And this would mean blood drawn, and also small bowel mucosal samples coming from the endoscopies with anesthesia and so on. So all these steps should be taken into consideration and were taken into consideration. And in pediatrics, we also have different type of problems, like growth in different groups age, especially the growth velocity, which is critical. It is crucial in some periods, as well as puberty and nutrition. So these are aspects that we do not really find, or we do not really care about. Maybe nutrition we do care, but we do not have the puberty issue and the growth velocity issues in adults. What about the ethics? A part of the ethical principles in general that guide research involving children, then in celiac disease, we would ask the question, would children and adolescents be willing to really intentionally eat, actively eat and constantly eat, for how long and how much gluten in the context of clinical trials? So it is the assent and consent issue here also. What about the target population? Which one would be let's say, the most in need age group of children or type of children to be targeted for a certain drug? We do have, as I already told before, those children who think they keep the diet, or who voluntarily have a little bit poor adherence to the diet, especially adolescents. We all know this age group is really difficult in time of compliance, and we really have to call them quite frequently for the follow-up appointments during our own clinical practice in our own hospital centers, because we know they need to be, in a way, to be supporting in really keeping the diet at this age. So adolescents would be one. But not only them, they are to high risk of persistent MASH2 with clit hyperplasia, or even MASH3 with villus atrophy type of small bowel injuries. So this is a target group. And then those children who are healed, they have maybe no symptoms, or they have mucosal healing, but they do have persistent symptoms. We do not really know if where this is coming from, but they really would also want less strict diet. So this would be a second type of children. And of course, this therapy as a complete, full alternative to gluten-free diet, but this is at this point quite far away. We are definitely not yet there. Okay. Now to special target groups in children related to primary prevention. Such group, of course, does not exist, or we are not thinking of it in adult age already. So this means children who for sure have some kind of genetic risk, or they have relatives, or I don't know, at least one index with celiac disease in the family, or some kind of associated diseases. And we would trial in the drug would, or an intervention of some kind, would try to prevent the development of the celiac disease autonomy, so to speak, to prevent the development and the appearance of the autoantibodies. And this is specific to pediatrics. And second are those children, the so-called potential celiac disease children. We are almost in some kind of conflict when let's say, propose a recommendation or give a recommendation to the parents and discuss with the parents what to do, because they usually have this positive serology with TG2, IgA positive, or EMA positive, or both positive, but they have absolutely no small bowel injury in normal histology. So positive serology in normal histology, and what to do. Usually, if they have symptoms, they go for gluten-free diet, but this is a potentially target group for novel treatments. And then when thinking of the study design, one should take into consideration the mechanism of action and what is the clinical indication for a specific drug, and which is the target population for that clinical indication and that specific drug, also keeping in mind the age-related indication. How to select the best inclusion and exclusion criteria for children in different age groups. This was really another very important problem. And do we need in all the trials the use of placebo? Because usually we need a very good control group in order to be able to compare the results between the drug arm and the placebo arm. And for how long, again, and how much gluten do we actively propose for the children to swallow in order to have the best and strong data to be really able to compare the two arms? And this is coming together with the acceptability of the participation. So many, many aspects to be thought about. I'm sorry, this is something, I don't know which is, what is the problem? Maybe sorry, shall I then, okay.
Probably you have to share again.
Yeah, this is clear, but I'm just wondering why is this needed to be shared again? Anyway, I think I was about, are you able now to see my full?
Yeah.
Okay.
Perfect.
Yes, and I was just about now to say that, of course, every single dietary intervention should be, of course, tested and proven effective. But then the question came, should all the phases of the clinical trial in children be delayed until after a total completion of the clinical trials in adults? Meaning, what about, let's say, the late phase of the studies like 2B and 3 phases of the studies? Do we really need in all age group of children and every single clinical indication and in all type of trials to really wait? So how to keep the balance between a little bit delaying and so in a way postponing, so to speak, the intervention and postponing the one available drug to be offered to the children, and to balance this to the safety, ethical, and sometimes wanting to wait to just see what would happen. So this was something that we should have to keep in mind, and it seems so that this was one of the most, let's say, discussed and not controversial, but let's say problematic and food for thoughts in all of this drug trial issue in children, especially because in celiac disease we do already have a treatment which is quite efficient, but of course we would not need to delay if a real good drug would be available. So this was an important question to be balanced. Okay, now I'm trying to go further, and this was working good. What about the outcomes? In children, don't we have some additional focused outcomes that may apply only to pediatric population? I was just talking about this puberty and growth velocity. And of course, mucosal healing for the point of view of the health professional is the main aim, is the main objective of the total issue of the drug itself, of the treatment of the gluten-free diet. But for the patients, it is also extremely important the patient-related outcome. They need to have improvement of the symptoms if they had symptoms in before, and they really need good health-related quality of life. So this should be also taken into consideration in every single type of trial. So to conclude, it's good to think about different scenarios, and this could mean different approaches and different outcomes. And of course, safety and ethics issues were to be faced. So because of all these topics and thoughts of this, let's say, task force of two groups, European colleagues and US colleagues, we really felt that a position paper was needed for the healthcare professionals. Maybe it's too much to say to offer guidance, but to offer our best thoughts and evidence-based findings that we were able to look around and to, in a way, be offered to the regulatory bodies, to the industry, to the patients, and to advocacy groups. And hopefully this was a little bit difficult to be followed, but I think you got the idea. I'm really waiting for your questions in the question and answer part of the presentation. Then I will hand over to Valentina, who is really maybe eager to show you the insights of the drug developments. I will stop the share now. Thank you.
Hello, thank you, Alina. And if Renata allows me, I would just continue.
Absolutely, Valentina, thanks.
And I was panicking because my presentation got completely messed up, but I found a saved backup for it, thankfully. So Alina already introduced the topic of today, and my duty today will be a little bit more to go into the different strategies that have been developed over time as possible targets for drug development in the context of celiac disease. So I will give you a little bit of an overview quickly of the celiac disease pathogenesis and the different therapeutic strategies that have been implemented or that could be implemented in the scenario of drug development in the context of celiac disease that are summarized in a review published last year in Gastro that I co-authored, and a little bit give you an idea of the tools that have been developed in order to implement also drug development and where do we stand now. So hopefully this goes through. Yeah, and just to quickly go back to celiac disease pathogenesis, in this setting, you all know that celiac disease is a very peculiar autoimmune disorder because it's driven by a dietary antigen, which is gluten. And we all know that typically it is characterized by a small intestinal enteropathy with a reduced bilous cytocryptide ratio and increased infiltration into epithelial lymphocytes, increased levels of specific antibodies, in particular anti-tissue transglutaminase and anti-endometrial antibodies. And we know that in order to develop celiac disease, the individuals need to carry the HLA, DQ2, and or DQ8 alleles, which are required, although not sufficient for celiac disease development. And this is also important for drug development purposes because only patients with specific genetic predisposition may be susceptible of certain specific type of therapies. So this is also something that should be taken into account when thinking of new therapeutic options for celiac disease. And the schematic that is from our review essentially summarizes the main steps of celiac disease pathogenesis that I'm going to go over quickly just to set up the scene for the therapeutic strategies that have been developed over time. So as you know, dietary gluten peptides in part escape digestion by luminal antigens, and so those peptides can cross the epithelial barrier, and maybe I can used a laser. I hope you can see my mouse instead. Maybe I can use this. Anyhow, I'll use the laser. I hope I activated the laser. I hope so. So the dietary gluten peptides that are undigested can cross the epithelial layer and reach the lamina propria, either the level of the lumen, as it has been recently shown by Ludwig Solid's group, or at the level of the lamina propria. They are targeted by tissue transglutaminase, which is a pleiotropic enzyme that has a crucial role in the pathogenesis of celiac disease, which is the deamidation of the gluten peptides. The deamidation of certain amino acidic residues really is key because it introduces some negative charges that can bond the HLA, DQ2, and DQ8 molecules that are expressed at the surface of the antigen-presenting cells. And this presentation is essential for the activation of a CD4-specific T-cell response that in turn amplifies the immune response at the level of the lamina propria by producing pro-inflammatory cytokines, including interferon gamma, IL-2, but also IL-21. Those cytokines have different functions. First of all, they contribute to amplify the T-cell response. Second, they promote the activation of B-cell response because we know that T helper cells may have a role of activating internal B-cell response that leads to the production of anti-tissue transglutaminase antibodies and anti-deamidated gliadin peptides antibodies that are then released in the blood, and that can be, of course, detected in the blood of patients. But also those cytokines can also contribute to activate the cytotoxic intraepithelial lymphocyte that resides at the level of the epithelial layer. And those cells, once activated, can interact with stressed epithelial cells that express high levels of non-classical HLA class 1 molecules or IL-15, other innate cytokines. And those in this interaction leads to then the damage at the level of the epithelium. So several of those pathogenic steps may be targeted by drugs that have been or are in the process of being developed in the context of drug development for celiac disease. And we know that gluten-free diet, which is at the moment, of course, the gold standard for therapy in celiac disease patients, needs to be strict and lifelong. And it has been shown that it leads to normalization of celiac disease serology and intestinal architecture in most patients. Although in some patients, we might still observe a persistence of an increase in intraepithelial lymphocytes, and some epithelial cell alteration may still persist despite the gluten-free diet. But what we have, we know that the resolution of clinical symptoms can be present in some, but not all patients. We know that there is a decreased quality of life in some patients because of the constant vigilance required for the diet. There is fears of cross-contamination. And also there are reports of gluten-free diet not followed up properly that may lead to lower intake of some nutrients such as fiber, but also nutritional deficiency. So those are some of the reasons why we might need alternatives to the gluten-free diet. And this has been also covered by Alina. There is the need, and this is more at the broader level, not only pediatric population, but also adult patients. There are reports that talk about incomplete remission up to 50% of patients despite a gluten-free diet with persistent symptoms in up to 40% of patients. We have a group of patients called non-responsive celiac disease, in which in some of them there is an additional etiology for the persistent symptoms, but in some cases this is due to inadvertent gluten ingestion. And then we have also a group of patients that do not really apply to the pediatric population, but in the adult population, we do have refractory celiac disease that develops in 1 to 2% of patients, and it is a very severe complication of celiac disease that might actually end up also into development of T-cell lymphoma in the gut. And even though this is not a condition that affects children with celiac disease, of course, this represents one of the areas in which drug development is actually focusing. And of course, a high occurrence and risk of inadvertent gluten ingestion in social life is definitely something that is very relevant for patients and represents one of the unmet needs for non-dietary therapies in the context of celiac disease. And this table from the review actually summarizes what are the unmet medical needs for non-dietary therapies as adjuvant of the gluten-free diet in celiac disease. And we know that one of the areas focuses on the possibility for drugs to resolve ongoing symptoms despite the gluten-free diet. So it's more directed to symptoms resolution. So for the so-called non-responsive celiac disease, some drugs are actually developed to protect from gluten exposure. Some others to facilitate healing, for example, rescue medications such as enteric budesonide. And of course, the holy grail for celiac disease, which is most desirable also for the pediatric population, would be to reestablish immune tolerance to gluten. And let's go back to our schematic of pathogenesis, because I'm not going to go into the details of each of the therapeutic strategies that have been developed, but just to give you an idea, we can summarize, and that's what we do. We decided to do in the paper to summarize the therapeutic strategies into three main categories. So one is the reduction of gluten antigenic load. So the reduction of the amount of gluten peptides that reach the lamina propria that can be presented by the antigen presenting cells. This could be done with different therapeutic strategies. So either think of genetically modified gluten or cereals that have a lower amount of epitopes, of gluten epitopes that might be recognized by the immune cells in the lamina propria. By sequestering antigens that are able to bind the gluten at the level of the lumen, reduce the amount of time that the gluten is in contact with the epithelial surface and then has the chance to cross the epithelial barrier. Enzyme therapies that are able to cut the gluten epitopes and so reduce the immunogenicity of those peptides, and the tight junction modulation, so reducing by modulating tight junction protein the permeability of the epithelium, the mucosal layer, thus consequently induce a reduction of the amount of gluten that reaches then the lamina propria. So these are definitely some of the strategies that have been developed over time. Probably the broader category is represented by the second strategy, which is immune modulation. So we can act on immune modulation at several levels. One of the most promising strategies has been represented by the inhibition of TG2, which is this enzyme that we know is not only important as an epitope, because the specific celiac-specific antibodies are directed against tissue transglutaminase, because as I said before, TG2 is very important for deamidating gluten peptides, right? So blocking the deamidation actually reduces the amount of peptides that are actually very prone to be presented by antigen-presenting cells. And so several companies have been developing inhibitors of TG2, but also there is another company, Japanese company, developed a compound, DonQ52, which actually blocks the presentation of the amidated gluten in the context of HLA molecules expressed on antigen-presenting cells to the T cells. And those probably are two of the most promising therapeutic strategies at the moment. There is a whole set of biological drugs or cytokine blockers that may act against IL-15, which is one of the most important cytokines in the context of celiac disease pathogenesis, although we know that in the context of refractory celiac disease, the success of this drug has been actually a little bit lower than expected, blocking T-cell intestinal recruitment, and also there have been studies more recently, some preliminary reports released on the effect of the anti-OX50 ligand in the context of the CD4 activation. And then in the end, of course, the induction of tolerance. The induction of tolerance is a strategy that would represent the only cure for celiac disease. The basis is the re-education of the immune cells to tolerate gluten as opposed to have enhance a pro-inflammatory response against gluten. And several companies have been developing essentially nanoparticles that include gluten epitopes, and they can then go to secondary lymphoid organs where they can instruct immune cells to develop a regulatory response against gluten. So divided, and this is another table that actually summarizes the different therapeutic strategies and options as I just summarized them before in the three different strategies and how they are applicable in adults and in children. As you can see, in children we have some controversies because some of the immune modulating strategies, of course, will have to be tested in the context of children, and as Alina pointed out, they are growing, they are around puberty in some cases. The immunosuppressive effects that might be a consequence of some of the biological drug might expose them to higher risk of infection, and especially in this specific category of patients where the gluten-free diet has been shown to be very safe and efficacious. Of course, it's very controversial for those drugs to be developed and to be expanded in the future. Of course, also for the tolerance induction, there are going to be some discussion because of the how many times the drug should be given, for how long does the tolerance induction last, and this is all something that is understudied and it's definitely not clear yet. I just want to point out that in the drug development path, there are a lot of different steps. Preclinical studies require different tools compared to clinical studies, and early studies different tools than late studies. And I just want to point out that some of the tools that are used in the context of drug development studies have been actually really informing us into also the pathogenesis of the disease and also the possibility to develop new diagnostic tools. So I just want to briefly mention some of the studies that have been performed in a preclinical stage of drug development, in which we were able to use either in vitro or ex vivo or even animal models to test new therapeutics. And this is very important from the early stages of drug development trials, but also a little bit where do we stand with the idea of gluten challenge and the other outcomes that we can use in addition to the most accepted endpoints of trials, which are essentially the storages and the PROs that we will talk a little bit before the end of the presentation. So just to mention that in one of the studies that we developed in Bana Jabri's lab a few years ago, in which we tested a compound that is able to block at the same time IL-15 and IL-21, we really use different in vitro strategies. We obtain cell lines from the intestine of patients, we obtain cell suspension of small intestinal epithelium, and we use organ culture. So different strategies in vitro that were able to be modulated. First of all, they were taken from patients, so they had the same genetic background of the patient, but also they were able to be used in the lab to be tested with or without the drug in order for us to test the ability of the drug to prevent the cytotoxic property of the cells. And this is just to give you an overview. This also allowed us to actually went a little bit into the details of the impact of the cytokines in the context of celiac disease pathogenesis. And we were able to show that both those cytokines are really required to promote the cytotoxic property of those cells. And this is to say that preclinical studies allow us to develop new drug, but also to gain insights into celiac disease pathogenesis. So it's a mutual, double-sworded in a positive way instrument. This is a paper published in Nature in 2024 by the group of Calvin Kuo and Mark Davis at Stanford, in which they developed these organoids that are mixed organoids with the epithelial cells and also the immune cells, and they were able to develop these tools that are actually very sensitive for testing therapy because they reproduce the individual genetic background, and they can also be manipulated with the addition of gliadin, peptides, but also with therapies. So this is definitively a tool for preclinical studies to test personalized treatment. And also in this context, this model allowed to unveil the role of IL-7 in the context of celiac disease. And this is another example in which, again, Baran Jabri and Valeria Abadi in Chicago, they developed the first mouse model that was able to reproduce gluten-induced villus atrophy. And in this context also, they tested several TG2 inhibitors that were able to prevent gluten-induced, as you can see here, shortening of the villi in the mouse, and also were able to restore the villus to crypt depth ratio. This was the first mouse model in which the villus atrophy was promoted by gluten ingestion, although it's a triple transgenic mouse model. But what is important here, that is another in vivo model for drug development in a preclinical setting. But when we go in clinical studies, so these are just tools that are used to develop new drugs and to test new drugs in the lab at an early stage. Later, we need to have some new tools that will allow us really to test whether those drugs are efficacious in patients. And so we really need to have some tools that allows us to test gluten-specific T-cell response in celiac patients on a gluten-free diet. And some of the questions are, should we give gluten? Should we test if those drugs are able to prevent gluten-induced damage? For how long we should give gluten? For how long we should give the drug? How to measure? So there are a lot of questions that will have to be addressed by clinical trials. And some of the outcomes that have been used in the context of trials are some tools that allow to quantify gluten-specific T-cell response, that are the ELISpot or also the tetramers and IL-2 release. And I will stop a little bit on the concept of IL-2 release. It is one of the most novel diagnostic tools for celiac. But also, of course, as we said, symptoms that are currently often called the patient-related outcomes, patient-reported outcomes. Histology, that could be, of course, it's in most clinical trials is used as a primary endpoint, so the ratio between the villus sign and the crypt depth, but also it has been developed more recently some alternative molecular morphometric tools in transcriptomic studies. And of course, what is the role of the blood markers? And it is especially relevant for the pediatric population, in which we are shifting into a serology-based diagnostic approach. And of course, gluten challenge studies are those gluten challenge is one of the tools that have been used in most of the double-blinded placebo-controlled trials for testing new therapies in celiac disease. And I won't go into the ELISpot, also for the sake of time. This was one of the first tools developed by Bob Anderson back in 2000, in which the production of interferon gamma was measured in response to restimulation with gliadin. But this paper published in 2021 in Gastroenterology, and this schematic really it helps me to give you the idea of how the gluten challenge could actually inform us also in the context of trial development. And so this was a study in which adults were randomized to have 3 or 10 grams of gluten, and then several outcomes were measured at 4 hours, day 6 or day 15, and the increase in IL-2 in response to gluten challenge was measured, symptoms were measured, also the production of interferon gamma by ELISpot, also tetramers were tested. gut-homing CD8 T cells were also quantified, the number of IELs and the level of histology, and of course, the morphometry was assessed. And so lower amount of gluten were able to only elicit symptoms and IL-4 at both 4 hours and after 6 days. But only 10 grams of gluten was able to promote after 15 days also the tissue damage. So it's really important to standardize those gluten challenge in the context of clinical trials. And I think drug development trials really helped us to this end to standardize the gluten challenge and really understand the timing of occurrence of the different symptoms, but also villus atrophy, but also other gluten-specific T cell type of outcomes such as IL-2 and interferon gamma. And IL-2 is one cytokine that has been brought up for the first time probably in 2019 in the context of the NextVax trial, in which it has been shown that after 4 hours of the ingestion of gluten, of the exposure to gluten, there is an increase of IL-2 that really coincides with the nausea and vomiting that were reported by patients. And after further developing the concept of IL-2 as a fast marker of response that actually is a proxy for the activation of the gluten-specific T cell response. Most recently, the group of Jason Tidin in Australia and Melbourne has developed an in vitro whole blood assay based on IL-2 release that could be performed also in patients on a gluten-free diet without oral challenge, but just re-challenging in vitro the cells of patients, re-exposing them to gluten. And this is some of the graphs from their recent paper that has been published this year in Gastroenterology, in which the IL-2 increase was documented in treated patients, but also in active patients, and this was much higher than of Colcerti control, but also non-celiac gluten sensitive patients with a very high sensitivity and specificity, and this was comparable to the gluten challenge induced the increase of IL-2 with a very nice, again, correlation between the whole blood assay and the gluten challenge induced increase of IL-2. So essentially, the whole blood assay reproduces the increase of IL-2 that is induced by an oral challenge with gluten. And this was also confirmed by the tetramer assay. This is just to say that this IL-2 release in the blood is really being implemented also in the context of clinical trials, although this is not standardized yet, especially in the pediatric population. And because it is not standardized yet, also in several years ago, I think this was 2022, there was a release by the Food and Drug Administration of a guidance for the development of clinical trials in the context of celiac disease. And essentially what the Food and Drug Administration said is just the two co-primary study endpoints should be clinical symptoms, so patient-reported outcomes, and intestinal tissue injury. There have been several comments to those guidance underlining really the fact that symptoms should not be considered as primary outcomes in the drug trials because are very subjective. And so a new guidance has not been drafted yet, but of course there have been several comments to it. And this is just one example from the New England Journal of Medicine paper summarizing the results of the anti-tissue transglutaminase inhibitor used for celiac disease, in which actually they use the villus-cytocryptase ratio as primary outcome and also questionnaire report-based evaluation of symptoms in celiac patients, so to adhere to the Food and Drug Administration guidance. But last year in gastroenterology, again, one of those reviews published in the special issue of celiac disease that was published in Gastroenterology, led by Elena Verdu and Peter Green. Ben Leubold from Columbia University published together with Joe Murray and a bunch, of course, of other colleagues, standardized recommendation for the design of clinical trials in celiac disease, in which they point out several very interesting tables that, for the sake of space and time, I didn't really summarize here, in which they report eligibility criteria, outcome measures, the parameters of gluten challenge that should be implemented in randomized clinical trials in patients with celiac disease. For instance, they essentially summarize two main type of scenarios: trials for evaluating the prevention of relapse in response to gluten challenge, in which participants should be adherent from at least 1 year to the gluten-free diet. And in this case, the gluten challenge should be carefully carried out with a FODMAP-free form of gluten. And then the efficacy of the drug should be evaluated using histology as a primary outcome, and as a secondary outcome, PROs, patient-reported outcomes. While for trials evaluating reversal of villus atrophy, instead participants should be enrolled with already a villus cytocryptase ratio below 2, and then the efficacy should be, of course, evaluated with primary histological endpoints. But this paper is also another paper that really fronts all the need of the different standardization for outcome measures, eligibility criteria in the context of trial, gluten challenge, and they also mention the fact that adults are one story compared to adolescents and children that require different trials. And I will go fast into the last part, because I'm going to my closing remarks, because Alina already covered the challenge for drug trials in celiac disease children. So we know that there is still the need of non-dietary therapeutic intervention also in children, even though the need is a little bit less than adult population, because generally the gluten-free diet is more effective in the pediatric population. But we need really to take into account that we cannot extrapolate the results from adult trials and not just apply it to children. Children have a different biology of the disease, different healing rate, different response to the diet. We need to, of course, take into consideration the role of the caregivers, the ethical issue that are associated with the length of the therapy, with the occurrence of the side effects, with immune depression in the context of immune modulation. And of course, we also need to consider in the pediatric population that we can think of prevention trials that are aiming at primary prevention in children at high genetic risk for celiac disease, but also secondary prevention to avoid the progression of potential celiac disease. This is definitely a unique scenario to the pediatric population. And again, the design for trials in children should be completely adapted to the different clinical scenarios and settings. And we still need standardization of oral gluten challenge, standardization, for instance, of IL-2 measures, and of patient-related outcome evaluation questionnaires that have not been standardized in children. And also this is summarized in this additional table that has been included in the review. And so to conclude, we want, of course, to underline that gluten-free diet is safe and effective in most patients, especially in children, that there are still problems with cross-contamination, quality of life, some nutritional issues, psychological issues that may affect compliance. There are still a lot of unmet needs for alternative therapies to the gluten-free diet, non-responsive celiac disease, refractory celiac disease, and that although several drugs are in the pipeline for celiac disease, the scenario is completely only targeting adults at the moment, that the main strategies that I summarized are reduction of the antigenic gluten load, immune modulation, and restoration of tolerance, that the main outcomes in therapeutic trials are histologies and patient-reported outcomes, although we really need to underline that because it's really a T-cell mediated disease, we really need to now take into account the importance of other biomarkers that are emerging, such as IL-2, both in the context of gluten challenge and in the context of in vitro gluten restimulation in the whole blood assay. And of course, as Alina brilliantly pointed out before me, the pediatric community the pediatric community needs really to understand and embrace that in the context of the pediatric population, we really need to drive the development of guidelines that differ from those of the adults, because we need to embrace the differences between adults and pediatric for the many different reasons that we just mentioned so far. Thanks to also the other co-authors of the review that we published, and also to our group in Naples and our funding sources. That's always good to thank. And thank you all for your attention.
Thank you, Valentina. Thank you, Alina, for your brilliant presentation. Very updated, and I think very important overview what is going on in the probably in the next years, what we will participate. We hope so. We have 3 questions from the audience. The first one is from Ilma Corponai-Zabo. If celiac disease is an autoimmune disease, what outcome could be expected by lowering gluten dose load? The immune system will see the antigen. I don't know which of you will try to answer to Ilma.
I think I can start because this is mostly related to the pathogenesis. I totally agree with Ilma. Probably for celiac disease, we need to have 0 antigen to really shut down the immune system, unless we were able to promote tolerance induction. Although, as we discussed a lot with Fritz when we were writing the paper, but also with Ludwig in other contexts, really memory T cells that are in the gut and are long-living memory cells are really there. So quitting these long living memory T cells that recognize even low amount of gluten will be very, very hard task also for the strategies that aim at restoring tolerance to gluten. So I have to agree with Ilma that probably this is a little bit utopic, but we will have to see where those tolerance induction strategies will go.
Thanks, Valentina. There is also another question from Chetil Stordal. Patient family comply well in their daily life, but ask for solution when traveling or going out for celebration. What is the challenge with getting glutinase to work, and what is the current status of such alternatives?
I think this is again for me. Katie, that's a brilliant question, because especially for the pediatric population now, what we
Valentina, we lose your audio for a few seconds sometimes. Now we don't hear you.
Can you hear me now?
Now, yes.
Okay.
Probably is a Wi-Fi problem.
Okay, so I repeat, because I don't know
Yeah.
until
Yeah,
when
yeah,
the
yeah.
connection was
Thanks.
good. I think this is a question that as pediatrician we are asked very often, and I think kids want and also their parents want a pill like the for lactose intolerance for lactose intolerance, in which they can just take the pill and go get a little bit of mozzarella, and they also would like to do that with pizza. But to be honest, we are not there yet, because unfortunately the amount of gluten that could be detoxified, it's not high enough for us to suggest it, to counsel them to really take gluten. So it could be just that at a certain point we will have some glutenases that will be taken just to avoid cross-contamination, but still in the context of a gluten-free diet and a strict gluten-free diet. So just, to a little bit kind of reduce the risk of gluten contamination in the context of, I don't know, for foreign travelers and something like that. But unfortunately, I think we don't have the power of allowing them to make the diet less strict.
Okay, one more question from the audience is from Dr. Yasin Sain. Is there any current or ongoing study about drug therapy in children with celiac disease?
I can start here again, but so probiotics, it's the only thing that has been really tried in children, and I didn't even mention that in my presentation. There have been some studies, such as Dr. Falk and anti-TG2, that have been ongoing in adolescents. This I know. In other countries, in Italy, there's nothing there yet. But in children before puberty, as far as I'm aware of, we're not there yet.
No.
Oh,
One question from me to Alina. Alina, what is your opinion? If we can have some lesson from therapy on diabetes and pre-diabetes children that we can translate in our field of celiac disease. What is your opinion?
I think we should, because there are a lot of similarities between the two diseases in terms of starting from the genetic backgrounds who are sometimes common in both children with diabetes and celiac disease. And I'm thinking maybe for the prevention studies, not yet, because in a way, when the serology is already there, when the antibodies are already there, I'm thinking out of the pre-diabetes, it's already late to intervene. So what would be the phase before, already before the antibodies, even, so as in diabetes, first are the antibodies, one of the three or two of the three or all three of them, and then the diabetes is coming at some point. But in this case, I'm thinking about this prevention study because if I'm not wrong, the only ongoing study is this probiotic who is targeting the prevention. I think it's aiming to prevent. But let's see, because in terms of pathogenesis, there were a lot of let's say insights about the viral infections and so on, who would be one of the, let's say, not the trigger, but sensitive point in terms of disturbing the immune tolerance on gluten. And this might be a common point with the newly published or recently published pathogenesis proposal and, let's say, development of vaccines for the children with diabetes. I'm now thinking on enterovirus and so on. And in celiac disease, there were publications looking into the viral infection. So maybe because of these similarities, yes, lessons from celiac disease and to diabetes, and from diabetes to celiac disease.
Okay.
Yeah.
Thank you. Last question is from Dr. Abdellah Radi. If we block celiac disease pathogenesis, I think we could develop other autoimmune disease and modulate other antigen tolerance.
That's a very interesting question. It depends on, I think, on the different strategies. For instance, if we do biologics, anti-IL-15, anti-IL-15, IL-21, anti-OX40 ligands, of course, we are blocking that not in a specific manner in the gut. So, of course, if those are involved also in other autoimmune diseases, more than enhancing other autoimmune diseases, I think we are blocking other inflammatory conditions probably. However, one of the main issues is not to block IL-2, which is one of the main growth factors for also regulatory T cells. So that's also something that is it's very tricky, because IL-2 is also is a growth factor for both T helper cells and so pro-inflammatory cells, but also Tregs, and so you want to be careful with that. We for sure are not blocking the whole celiac disease pathogenesis, so we're not really immune depressing the patient in any of those strategies that I just mentioned. The reinduction of tolerance should only act on gluten, because the epitopes that are provided into the nanoparticles are just the gluten epitopes. So I theoretically we should not. And but if we want to see something more broad, also to connect to what we were saying about celiac and type 1 diabetes, some strategies such as the teplizumab, which is an anti-CD3, that's blocking or ideally, the idea is that it's delaying the onset of over-diabetes in patients without immunity and with antibodies that are in a pre-diabetic stage. We might think that this strategy might also prevent preclinical celiac from developing into celiac, but I don't think it's justified at this point to use it yet because anti-CD3 is even broader because it's an anti-lymphocytic, an anti-lymphocyte biological. So we will see with the teplizumab results if they will have also secondary outcomes in which they will look into preventing celiac disease onset. We will see, I think.
Okay. Thank you, Valentina and Alina. Thank you for a very interesting talk, for your answer. And I think we are in time. We say bye-bye. Bye-bye to everybody.
Thank you. Thank you, Renata. Thank you, Alina. Bye, everybody.
Bye.
Bye bye. Good evening. Bye.